Thank you. That had more to do with my age probably than anything else. But you know, if you stay in this business long enough, you'll get some things done. So, but thank you very much. That's very kind of you.
So we'll start with arguably the most timely question. So you're currently your research is focused on COVID-19 currently, and you have one of the few BSL3 laboratories in the nation. Can you talk about what you guys are doing right now in your lab?
Sure, be happy to. It's almost easier to talk about what we're not doing, but as you can imagine, everyone's lives has changed in many dramatic ways, as has mine. This is my third pandemic. I worked the first SARS coronavirus pandemic in 2002, 2003, and did that for a number of years, and then the 2009 H1N1 pandemic came along, and I worked that one. And when this one hit, because of my experience in working with some of these, and actually there's very few people that work with coronaviruses or have, I really felt compelled to get involved. But one thing I've done differently this time is taken lessons from the previous pandemics in terms of what were kind of the difficulties in terms of getting an emerging program built, targeting a new virus that no one knows anything about. Really, right now, my efforts can be divided into three areas. I developed, my laboratory fairly quickly developed a high throughput screening platform for looking at FDA approved repurposed drugs that could be used in the current pandemic. As we all know, it takes many years and many millions of dollars to get a drug from inception to to market. So the only way we're really going to find drugs for this virus is to take things off the shelf, so to speak, and apply them. And so working with a large computational biology group here who actually do the biggest high throughput screen, albeit in silico analysis, we have been screening FDA approved drugs for their ability to inhibit viral replication in some manner. So I work in that space right now and have a team that's doing that work, led primarily by an MD PhD student in my group. In addition to that, we all recognize the importance of vaccines, and I have been working with a large group of immunologists who are also in collaboration with a number of vaccine companies. And we're currently testing vaccine candidates in animal models for the preclinical evaluation and down selection of vaccine candidates to the ones that will go into phase 1, phase 2 clinical trials, hopefully, by the fall. And then the third area that I'm working with is just more of a conceptual landscape of identifying and developing models that can be used for understanding the SARS coronavirus 2 biology and the mechanisms leading to pathogenesis. And that I do with a large number of collaborators throughout the School of Medicine and the UAB kind of university area. It's a large university. We're actually the largest academic medical center in the country with over 1,400 beds in our hospital. So this is a huge campus and a huge institute. We rank, I think now 21st in the country in NIH funding. So there's a lot of people here who have ideas about, what we can do to help treat patients with COVID-19. And so I work in a consortium of investigators to help develop those models. Anything that needs and requires live virus work really ends up in my lab as the only lab on campus that has capability of handling the live virus. So that's the short answer in terms of the space that I'm working in. And any given day is kind of a different focus. And then we're also having young investigators from other laboratories that essentially come to work with me in kind of a borrowed manner, if you will, to work on an area that they're passionate about. So we do that. And that's how we're able to do so much in a short period of time.
So if you'll allow me, we're going to digest that all a little bit. Okay. So you've got 3 major teams. How many, essentially how many scientists do you have on each? And what is you mentioned the one PhD, MD PhD student that you have spearheading the computational work. How many other graduate students do you have in these efforts?
So that's an excellent question. I have currently, ironically, when the pandemic broke, I was in between kind of phases of my lab. I had just finished an R01 on flu. I have a new R01 starting next month. I had several postdocs that had kind of gotten to the end of their postdoctoral training, and they have moved on and found other career advancing jobs here at the university or at other places. And so I actually had a small lab. When all this broke, it was me and one senior technician. And For weeks after that, I've had to get back into the lab. I spent Tuesday night, Wednesday morning, all night in the SEB lab, in the BSL3 lab, in the spacesuit collecting time points. So that's been one of the ironies of this period, is I've actually gotten back to the bench far more than I have in, say, 20 years, which has been a great kind of rejuvenating activity. But in terms of the teams I have about 3 to 5 PhD, graduate students or PhD postdocs working in the lab on the drug screening platform. The vaccine platform is really being done by 2 senior research techs that I have in my group. And then the animal modeling project has about 4 graduate students working on it now. Those 4 are actually not working for me. They work with somebody else, but I do sit on their PhD committees. Their bosses wanted to get into the COVID-19 field. And so they actually kind of work in my lab primarily. I mean, I see them more than their other lab sees them. And they work in that. And so I think what's very different about today than, maybe it was when I went to graduate school is, a lot of young people, they need kind of this big world, big kind of environment kind of setting. When I went in, you found a lab and a mentor, and you worked in that lab, and you shut the door and you just worked away. And the only time you saw other PIs was maybe a committee meeting, or if you need to borrow a reagent or maybe ask for advice. Nowadays, it's not like that. Graduate students are really kind of their own person, and they're in charge of their own dissertation. And so they really kind of work freely with a number of other investigators. And while they have a home lab and a place they can call home, and a mentor that takes care of them and looks after them on a daily basis, many of the graduate students here work a little bit more freely than that. And that's actually a very rewarding kind of experience, because I get to kind of increase my mentoring footprint, if you will. And that's a bad way of saying it, maybe, but on a number of other graduate students, I get to help train them as well. And they get to learn from multiple investigators, because we're all a little different in that regard. So that's kind of the size of the team there. So not terribly large in the grand scheme of that. There's many labs that work in the space I work in, and that's 75 postdocs, kind of environment, which I trained in. I know what that's like. But I also know that in those environments, there's generally a handful of people that are really the ones kind of getting things done. I try to work in more of that focused approach.
Maybe to your comment on footprint, I imagine you are now officially a part of a very large group of scientists, scientific trees or family trees. Can you, for the animal modeling work, can you describe some of the models you guys are using?
All right. We're actually using here at UAB, because of the facilities and some of the limitations that we have in our facilities, in our BSL3 facilities, we're mostly working with hamsters and ferrets. I was trained during my postdoctoral years. Right after I left UK, I went to Children's Hospital in Cincinnati and learned how to develop transgenic and gene knockout mouse models. That was at the early days of that field, and we were just then learning how to make a knockout that was specific to a certain organ system. That's fairly commonplace, or even a specific cell line now. But when I was making that transition to my postdoctoral training, that was a brand new thing. So I was steeped in mouse modeling of diseases for the first part of my career, only to find out that many human viruses don't infect mice very well. Viruses, unlike bacteria, fungi, other types of pathogens, are often very species specific, and none more so than probably coronaviruses. So I had to learn to adapt. If I was going to study viruses in animal models, you have to find the animal model that's going to best recapitulate the human disease. And as nice as mice are, because of the wealth of reagents and the genetic backgrounds and the genetic manipulations that we can do. It's not always the best model, and this is what we're struggling with in coronavirus. So we're working with that here. I spent the early part of the first SARS pandemic working on non-human primate models, because at that time, that was the only animal model. But that was at another place, and they had a large primate colony as part of that institute. And we were able to do that. And that work requires, teams of veterinarians to be able to do that work. Non-human primates are not easily worked with. Let's leave it at that. But so I have worked with, mice, hamsters, ferrets, non-human primates. I did some PhD work there at UK using rat models. So I've kind of run the spectrum of that. And each species brings its own unique challenges, not just in terms of animal handling and husbandry, but also, the types of disease manifestations that you develop. And probably one of the hardest things is, unlike humans, we get sick, we go to bed, we lay down. We don't worry about predators. Animals do. Animals that live in the wild do, even if and so animals are very resistant to showing symptoms of disease. They can't afford to, because then they become food for something else. And so one of the biggest challenges that we have is developing animal models that have the symptomology that we see in humans. An animal thing out in the wild is just a signal for some other animal to come eat it, probably. So, these kinds of things make very, very unique challenges. So I have done that through the years. Our work currently is really just in hamsters and ferrets, but they have their own unique challenges with regards to understanding COVID-19.
What are some specific signs that you're looking for in these animals?
Well, we take a clue from, what our physicians, I work with a number of physicians and physician scientists being in a clinical department. We generally take a clue. I work with, because my department has, in addition to anesthesiology, but also perioperative medicine, those are physicians that are intensivists or critical care docs. And in many large academic medical centers, that is a physician that is in charge of a patient undergoing a surgery, whether it's a hip replacement or a gallbladder removal or, a heart valve replacement or what have you. The anesthesiologist in our hospital is your doctor, and the surgeon just comes in and does the surgery and then leaves. But the anesthesiologist and critical care intensivist is actually your physician from beginning to end and sees you all the way through that critical period. So we see a patient as undergoing a procedure as at some point a critically ill patient. We're probably keeping them alive at some point in time on on an anesthetic, on a ventilator, or what have you. And so many of our critical care docs are now critical care COVID-19 docs. And so I interact with these folks daily, and they tell me what they see in COVID-19 patients. I take that information and go back to the bench, bedside to bench kind of mantra, to actually develop animal models. So to your question, we're looking for an early signs, upper respiratory signs, rhinitis, congestion, runny eyes, things of that nature. But also some animals will cough. Ferrets actually cough. It's more like a hack and a gag than a cough, but it is cough. It's what's happening. It's just the way it sounds is different. We're looking for signs like that. We're also looking for symptoms such as, sorry, I said signs. Those are symptoms. For signs such as body weight loss, body fever. We use telemetry devices often to determine fever in animals. Some animals, importantly, they don't develop fever. They actually lose body temperature. That's how they decide. That's how their bodies have evolved to fight viruses, is they just reduce the temperature of their body so the virus can't replicate, whereas we increase our body temperature to fight the virus better. So those are the kinds of things that we're looking for. Beyond that, we develop X-ray and CT imaging capabilities and things. These are all the types of ways that we can determine disease in animals to SARS coronavirus 2.
How do you feel that the environment of a College of Pharmacy versus College of Medicine in terms of research and clinical opportunities has equipped you for this?
That's an excellent question because I'm pretty far removed from what many of the alumni at the UK College of Pharmacy went on in life to do. Um, it's a long story how I got into viruses, but suffice to say that I left UK and went to join a postdoctoral fellowship at Children's Hospital in Cincinnati at the height of the gene therapy days. This is when we were going to give genes as medicine, and it was just going to be that darn simple, and we're just going to fix everything, especially all these congenital genetic diseases. And I started my postdoc the day after Children's Hospital shut down their gene therapy trial for CF after the first two patients got sick from the adenovirus vector that they were delivering to deliver the CFTR gene to patients with CF. And so my very wise postdoctoral mentor said, well, we're never going to fix this gene therapy business until we fix these adenovirus vectors. So you young lad are going to go study adenoviruses and adenovirus vectors and see if we can find an adenovirus that'll deliver genes better without making our patients sick. And so that turned out to be the height of the gene therapy days. The day I started my postdoc, from that point on, it crashed. And I thought, well, darn, this is nice. I just picked a field that's going to be gone in 5 years. What the hell am I going to do with myself now? And so I actually, from that point, realized that, you know what, Harrod, you don't study gene therapy, you study adenovirus pathogenesis. And viral pathogenesis is probably a really important thing because we don't have any drugs for viruses. We have lots of antibiotics for bacteria. So that started me on the 25-year odyssey that I'm on now, which is to study viral pathogenesis and viral disease and to try and help move the needle a little bit in terms of viral infections in humans. To your question, how did this prepare me? Well, by going through a pharmacology program and understanding the importance of drugs and things like that, I have continuously, at every aspect of my career, reached back to my understanding of pharmacology and how drugs work to be able to discern whether the targets I'm finding are actually druggable, if you will. If the drugs that I test for vaccine companies and antiviral companies and what have you actually meet kind of this concept of this is a drug that we could actually give people. There are lots of people who come to me and they want me to give some crazy lipid peptide combination to kill a virus or whatever. And I'm like, yeah, that's great, but how are we going to put that into a pill? How are we going to put that in an injectable to be able to give people? So that pharmacology basis actually permeates all fields. If you're going to be a translational scientist, like I strive to be, you're going to have to understand that in part of that translation, you're going to have to understand the pharmacology that's involved with making drugs and interventions that are useful and usable in individuals. So the training at UK was fantastic in that regard. I was steeped in medicinal chemistry, pharmacology, safety pharmacology, drug metabolism, all these things that I use on a daily basis, because many of those concepts aren't just useful for drugs, they're also useful for other biologicals, including our endogenous biologicals. Understanding the half-life of a protein, of a cytokine in the blood or something like that, all goes back to me learning about the half-life of drugs and metabolism in safety pharmacology and things that I learned at UK.
How do you feel the clinical opportunities in these two different colleges differ, and essentially why College of Medicine if you're a pharmaceutical scientist?
Well, that's an excellent question. You know, one of the reasons is that, you know, UK is very unique in the sense that it has this very large PhD program and this kind of research footprint compared to many colleges of pharmacy. And so you don't find that in too many other places, and you guys probably know this better than I do anymore. And so I think the thing to think about in your careers is not just think about, you know, pharmacy schools versus medical schools or things of that nature. They're really all the same. You know, I could have ended up in government labs. In fact, you know, I've almost ended up in government labs at different times in my career. Government labs are nice. They don't run out of funding. They have their own funding limitations, but they don't run out, and they don't have to write grants necessarily to get that funding. So, I mean, every environment is different, but when you come back to it, the concepts are all the same. We're here to help people. We're here to develop interventions. We're here to study the biology of human disease and all those kinds of things. And the environment around you is important in terms of what it can give you for support and knowledge and you know, enthusiasm for science. But we need to not think in terms of silos. One of the things I liked about UAB in terms of this massive medical center is the fact that there is no there's no silos, right? People don't work in their department. I'm in a clinical department of anesthesiology and perioperative medicine. How a virologist into that is probably a conversation best had over beers. But short answer is clinical departments have money, and because they're taking care of patients and they have a huge revenue stream. Basic science departments anymore do not. And so, it's really difficult at times when you need $15,000 for a new piece of equipment because your piece of equipment just died. It's easier to do in a department that makes $87 million a year versus maybe one of our basic science departments that makes $7 million. And so it's much easier to go ask for that $15,000 piece of equipment that just died. It's critical to your research in that setting. Plus, I just wanted to get back to closer to translational research, and that means being around patients, and being around patients means being around physicians. So for that reason, if there was a College of Pharmacy here at UAB, I'd probably be a part of it, but there's not. The pharmacy school's over at Samford, which is, 5 minutes down the road. And I actually have pharmacy students that come see me all the time, who somehow stumble across my CV or my bio sketch or whatever, and they see, oh, he trained in a College of Pharmacy. They'll go talk to him. They'll come in here in my office and we'll have a chat about career opportunities and things of that nature. So I don't really see the differences between that, but that's just me personally. If I came to the UK College of Pharmacy to set up a research operation in a shop, I'd be working with College of Pharmacy faculty as well as being across the street over to Chandler Medical Center working with those folks as well. In fact, one of my best friends and frequent collaborator just stepped down from Chief of Pulmonary Critical Care Medicine at UK. He's now Vice President of Research in the Department of Internal Medicine, Peter Morris. And he and I talk about all the time, about all the things going on at UK. And of course, the College of Pharmacy is, one of the bright shining stars of UK's, umbrella of healthcare. So it's well recognized, and I would encourage people to think outside your department, outside your college, and look to see what's out there because there's lots of opportunities.
Yeah, I think we are uniquely advantaged just to be so near the clinical setting, and just the number of collaborations we have. Why do you think that drugs for viral diseases are so scarce? Is it just a difficulty issue or a lack of interest?
An excellent question, and one I have grappled with for almost the entirety of my career. I think partly it's just the difficulty of working with viruses. Unlike bacteria, if you find an antibiotic, you can test the antibiotic on a bacteria sitting on an agar plate, and that can be screened very rapidly. Viruses require a host, and the host often is us, but it could be any other animals. It could be plants for that matter. There's obviously viruses, huge amounts of viruses that infect plants. Because of that, it just adds a complexity to that system. I think that's part of it. I think part of it is just the sheer, I'll call it fear. People don't like working with viruses. Viruses scare us. The word virus scares us. They make movies about being scared about viruses, right? I don't know of any movies they make about being scared of bacteria. I just don't, right? Anytime you see a Hollywood movie about an outbreak, it's always a virus, right? And so I think partly that's because of the difficulty of that and what have you. Also, viruses require so much. I mean, they're so small. The influenza virus has 10 genes, 8 gene segments. And it's so simple. It uses so much of the host. And so when you make a drug to interfere with the virus, you almost are making a drug to interfere with the host as well. There's a few exceptions. Obviously, Tamiflu is a hemagglutination, neuraminidase inhibitor. So that's a viral mechanism. But we have neuraminidase enzymes in our human genome, and we use neuraminases all the time. It just so happens that for whatever reason, that drug seems to not affect our neuraminases. But often the drugs interact with the host on a great deal. And so there's a lot of toxicity with that. And drug companies are very risk averse. And so making a drug that has some level of toxicity, even when you recognize it on the front end, just probably stops a lot of it very early in the preclinical stages in the in vitro with basic scientists like me studying a drug in a cell system. So I think that's part of it. I think if one of the things that comes from this COVID-19 period is we're going to kind of get over that and realize, we got to fight through that somehow and make better drugs. I think that's part of it. The other issues were just technical issues that I think have made it difficult for people to work with viruses. So I think that's part of it. I think I mean, we've discovered, a number of FDA approved drugs that could be repurposed for viruses, and I suspect they'll work against some other viruses as well. I'll throw one out there, Prozac. Prozac has SARS coronavirus 2 antiviral activity. Who would have thought that? Well, our computationalists discovered it. They told me, hey, try Prozac. We did, it worked. And so, there's probably lots of things out there that can help. But I don't see Prozac as an antiviral drug. I see Prozac as a prototype, once we understand why Prozac is working, to develop other drugs against it. Hopefully drugs that don't have so much of the neurological, aspects that Prozac might have. I mean, we don't really want to kind of get everybody, all Prozac out for, their antiviral therapy, although it may not be such a bad thing. But nonetheless, that's the way I look at a lot of these. But it's very difficult to understand sometimes why. But I think a lot of it is the pharmaceutical industry and the risk adverse aspect of that.
So talking about some of the difficulty working with the viruses, you're at one of the few BSL3 equipped labs in the nation, and you also helped develop the BSL3 lab at Loveland's Respiratory Research Institute. Can you talk about some of the process that goes into setting up that sort of laboratory?
So I built 2 facilities, and I say built, I mean, I sat with architects and designed them, and this is the way you walk in, and this is the way you walk out, and here's where you put on the spacesuit, and here's where you take off the spacesuit, and things like that. I didn't swing any hammers, so I like to say I helped build 2 facilities, and then I've helped this facility here. When I joined UAB, almost 6 years ago now, they had one of the 16 regional biocontainment facilities built by the NIH after 9/11, but it had largely set unused and I dare say dormant. There were some TB folks that were using it, but TB kind of falls in a little bit of a unique spot for BSL3 work. It's not considered too much of a it's not a scary BSL3, not like SARS or flu, bird flu and things like that. So, those facilities require a great deal of oversight. You have to have dedicated personnel that oversee these facilities, because there's just layers and layers of protection on you. People have asked me, I've been on the news a few times here in the last few weeks, and people say, well, what's it like working in a BSL3 facility? And I was like, well, geez, it's safer than being at Walmart these days. I mean, we just have so many layers of protection. Generally speaking, I've taken on a few techs and graduate students that were like, geez, Dr. Harrod, I don't know if I want to go work with that. And I'm like, well, we're not going to force anybody to do anything. I don't want anybody to go in there who doesn't want to be in there. Over time, people come very comfortable with all the layers of protection, and they realize that if you just kind of follow the protocols, there's very little that can happen to you. But it's really kind of understanding all the different layers of protection. One of the biggest protections we have is just an enormously elaborate ventilation system within the facility. It's always pulling air away from you. We're always pulling air away from you, so that if the test tube you're working in spills, it's all blowing away from you. And just like a laminar flow hood, right? That's a very, very important part of working with highly pathogenic organisms and things like that. In addition to all the other layers of protection, I call it the spacesuit. It's not a spacesuit. It's 5 or 6 gowns on top of one another with all this PAPR system and an air ventilation system blowing around your head and what have you. So, you just kind of have to sit down and understand these things. Where this really gets to be difficult is when somebody says, hey, we're going to turn this lab down the hall here into a BSL3 lab. And you say, boy, that's going to be really tough, because it needs its own dedicated ventilation system. It needs its own dedicated entry and exit procedures. It needs a room around a room around a room and things like that. So you have layers of protection in each of the rooms. Autoclaving facilities, in our BSL3 facilities, nothing comes out that either one isn't incinerated or 2 autoclaved. So there's a lot of things that go into that, but once you build all that stuff in, it's not terribly difficult. And I would dare say that most of the folks that have come to me and have been reluctant to work in that environment, after a while, they actually kind of enjoy working in that environment. There's something cool to be said when you go to cocktail parties or you go home and hang out with your family. And you're like, yeah, I was in the spacesuit yesterday working with the SARS virus. Wow. So I think a lot of people find an excitement in it, and you know, it's obviously very important. I mean, we work with these pathogens in these facilities because for a reason. They're really dangerous, and they're really important to study, or we wouldn't take on that complexity of that danger, right?
So this is your third pandemic. Can you talk about some of the lessons that you've learned in the previous two that you have been able to apply here?
Ooh, that's an excellent question. Lessons. I think the lesson we finally got this time was to do your homework and develop your protocols before you get started. So, and I don't mean just protocols in terms of here's how you infect cells with the SARS virus and what have you. Here's how you do the plaque titer assay, which is a way of counting viruses. Here's how you do your PCR. The protocols and procedures I'm talking about are what we call our SOPs, or standard operating procedures. And these are protocols that are vetted, meaning they're evidence-based to work, but also to provide safety for not only just doing the studies, but actually taking samples out of the BSL3 facilities. We do have samples that come out. The samples that come out are either isolated RNA, formalin-fixed tissues or cell monolayers on dishes, you know, 6-well plates or what have you. Heat killed, so the SARS coronavirus is actually fairly sensitive. I say fairly, not a lot, but fairly sensitive to heat. So if you can heat a sample to 70 degrees for 30 minutes, you're going to kill the virus that's in that sample, things like that. And we did all our homework up front to get all these procedures in place so we could get samples out and analyze them, because you can't take every piece of equipment you own. You can't take that $700,000 flow cytometer and put it in the BSL3 lab, you know? And so being able, so you're going to increase your research throughput by developing procedures that allow you to get things out of the facility and analyze them through standard means. So I think one of the biggest lessons we learned this time was do your homework on that first. While you're still trying to figure out how to grow the virus and things like that, you can do things to kind of prep yourself for speed later. So I think that was a big concern and a big benefit with this one. Secondly, the lesson I learned from the first two is everyone's starting from zero. Everyone is largely ignorant of what to do. And don't be afraid of the findings that you have that seem counterintuitive to what other people are seeing. Case in point, and I learned this somewhat with the second pandemic, the H1N1 pandemic in 2009. I had an NIH, large NIH contract to do vaccine testing for bird flu, H5N1. And the first two patients with pandemic H1N1 got sick in San Diego. There were two children in a pediatric center in San Diego. And the NIH in our weekly teleconference with them said, hey, we want you to stop all the H5N1 work. We want you to shift to H1N1. And we're shipping you a sample of that virus today. This was 3 weeks after the 8-year-old child died of H1N1. And we started growing it. And I had a very, very talented junior scientist, junior faculty member who had actually worked at St. Jude's in the big flu group there led by Robert Webster. And we're growing the virus one day, and in one of the weekly teleconferences, I said, yeah, we grew the virus and we're about 10 to the 8th PFU per mil. And there's dead silence on the phone. And they said, okay, that's great. It's good to hear. Let us know, keep doing what you're doing. Let us know when you have, these assays set up or whatever. Well, about 3 hours later, the CDC calls me and they say, tell us how you're growing the virus. And so we had that conversation. And then, the next day USDA called me, which is a big bird flu group. So they had been mobilized in this. And they're like, yeah, we understand you guys are growing the virus quite well. And so for reasons that I still don't understand, we were growing, well, I do understand, but for reasons at the time I didn't understand, we were growing the virus at least 2 logs higher than everybody else. And, that's for someone who really never trained in a virology lab. I mean, if there's one thing kind of sometimes that catches up to me is you're a virologist who never trained in a virology lab. You just kind of self-taught. Well, not really. I actually hired a lot of really smart virologists from other virology labs, but it's a different story. So it turns out we were just using a different type of trypsin. Influenza requires trypsin to infect cells. And we were just we screened several trypsins to find out which ones would give us the best, and no one had taken the time to do that yet. So don't be afraid of your findings. And if you've repeated them and found them over and over, you're probably got something that nobody else has. So don't be afraid to stick your neck out there and say, hey guys, I think you can do it better this way, or I think this is a better animal model, or this is a better way to grow the virus, or what have you. And so I think with this pandemic, I certainly have a lot more confidence going up what I call the big boys and girls in the virology world with ideas of things. And we've already made some headway in that in terms of using primate lung cultures, which is maybe what my claim to fame is nowadays, is I use rejected lung transplant material. I take those human lungs, I isolate various cell lines from those, and then I'm able to grow these little lungs in a dish kinds of things. And I study viruses on that. And that's really hard to do, and not a lot of people do it. So I actually am, kind of sharing with World Health Organization, other organizations, some of the findings that we're having in these types of model systems. It turns out that this virus actually doesn't replicate nearly as fast as, like, say, flu does in those same systems. So this early idea that this is some wild, rampantly growing virus in our lungs actually isn't true, not compared to flu anyway. So we're, those kinds of things, I think, and I think just the experience of kind of going through the unknown with other groups has helped a lot with this pandemic.
I feel like we've just scratched the surface scientifically, but we do have just a few minutes left. So I do want to shift towards some of your personal professional development.
Okay.
Can you describe some key turning points or decision points that have shaped your trajectory to be what it is?
Excellent question. So I think the first was my transition from my UK years to Children's Hospital in Cincinnati, where I did my postdoc. And one, at that time, I was sitting in a seminar about a year before I graduated UK with my PhD, and investigator came down from Children's Hospital and gave a seminar to the College of Pharmacy, and he described, Dr. Jeffrey Whitsitt, he described this new technique for developing transgenic and gene knockout animal models specific to the lung. And I read this and I said, wow, mice are now our petri dish. A living mouse is the petri dish. You can put any gene in, you can take any gene out, and you can turn it on and turn it off when you want. And I thought, I got to know that. And what I have learned through my years is that every 5 to 8 years, there's a revolution in science. There is some new technique. Right now, it's CRISPR, right? And maybe some minor ones. Before CRISPR, is stem cells. Before stem cells, it was microarray. Before microarray, there's these cataclysmic shifts in our ability to do things that come along at different times. And I think one of the things I learned in that transition was don't be afraid of those. Don't be afraid of trying something new and doing something new. And if you look back at my bibliography or what have you, you'll see, hardcore computational bioinformatics in the mid 2000s. Nobody was really doing that. And I went out and hired 2 really smart postdocs that were good computationalists, and they taught me how to do it and things like that. But don't be afraid to take on these kind of cataclysmic shifts in our ability to do things. It always makes you topical, and it always gets your grants a second look. Like, ooh, a new technique here to maybe even answer an old problem. That's a cool thing. So that's one. Secondly, do things that nobody else is doing. Look at whatever field you are, whether you're in liver biology or neuroscience or whatever. Identify the things that people aren't doing. Go up and ask the really smart, important people to say, hey, what do you think's missing here? What do we just have no idea about, right? And where are our gaps in our knowledge? And go fill that, and find a lab that's trying to fill that, and go work with them. And that's a big thing, I think, anymore, because if you just follow the fields, especially as a young investigator, you're always going to be overwhelmed. It's you against some massive lab with 5 R01s and 3 floors of a research building and God knows how many people working for them, right? You just can't beat that. So don't. I think that's a really important area. And then lastly, and this does not make me popular with some of the other professors around here because their postdocs end up on my couch trying to figure out how to get their career. When you get to your postdoc years, absolutely work with your mentor. Find a mentor that's willing to give you a project that you can take with you. Because people ask me all the time, how do I get a faculty position when all my job responsibilities are aimed to of my boss's grant? You're not going to get a faculty position off that. That's your boss's work. And so you have to go into those postdoc junior faculty training years to say, okay, Dr. So-and-so, I'm going to do your aim too, but I want to carve out a niche that I will work on, whether it be nights and weekends or whatever, that I can take with me. I need a project I can take with me, because when you go to give that seminar for that faculty position, those people hiring you are going to know, what are you going to do here? Because you're not going to do aim 2 of your boss's project. So what are you going to do here, and how are you going to develop your career? And if you can't get that out of your postdoc mentor, don't be in that lab. And so I tell this people all the time. And then about 15 minutes later, that postdoc mentor comes down to my office, like, what are you doing telling my postdoc this stuff? And I'm like, well, they asked me how to get on in their career, and I told them how I would do it. And that's how I did do it. My postdoc mentor told me early on, this is your project. This will always be yours. As long as you work for me, I'm the senior author, but you need to write the K grants and the small grants, the F grants and whatever, to move this on. And then you'll take it with you when you leave. And we'll figure out a way to collaborate and do things. If I need stuff from you, we'll figure that part out. He wasn't an easy individual to work for, but he was very fair in that regard. And you gotta find that. So you have to find projects that are movable with you. And your PhD years, your grad student years, that doesn't matter so much. I went from pulmonary vascular disease to airway epithelial biology from graduate school to postdoc. I stayed in the lung, but I switched from the blood side to the airway side. Didn't hurt me one bit. So don't worry about what you're doing right now. Just get your papers and show that you can get things done, and prove to your postdoc mentors that you should be hired because you're going to get things done in their lab. But after about the second year of your postdoc years, where you kind of increased your skill set and your critical thinking skills and your writing skills, then you're going to start thinking about that transition to if you want an academic career. The last thing I'll mention regarding that is, there are many, many fabulous careers, enjoyable, rewarding careers that aren't about being a faculty academic scientist slinging grants out the door every quarter. Many careers. And most of the people I have trained did not go on to be like me. I have one that works for the CIA, and she can't tell me what she does because she works for the CIA, but she does tell me, you're okay, we don't investigate you. I think what her job is, based on the questions she asked me, is she works for the CIA reviewing classified documents of scientists in other countries to determine whether they have the ability to develop biothreat weapons to create, a synthetic SARS or whatever. And so she basically informs, she uses her virology and biomedical skills to be able to say, yeah, I don't think that scientist could make a virus, or yeah, that scientist good. He's got all the right centrifuges, and he bought a rotor for a centrifuge, and that's a centrifuge that we use to make, to isolate viruses or whatever. So there's that kind of job. I have many investigator, former trainees that are in national labs, and they just do what the national labs need them to do, and they're advancing through their careers. One is a tenure track scientist in the Department of Pediatrics at University of New Mexico. He basically does all the genome sequencing of unique patients, of pediatric cases that have unique disease, that have congenital disorders, and trying to figure out why this young child has this condition, and he's sequencing the patients. So there's that. I'm just trying to think. I have one that's working at USAMRIID on Ebola, one at USDA. So these are all folks that don't write for grants. They go work in laboratories. Two went on to be lawyers. One quit to be a professional cyclist, and they used to race right behind Lance Armstrong for years. So, I mean, there's all kinds of trainees that end up doing a lot of different things, and you just have to decide what makes you happy. But you're not destined to a life of writing grants and fighting with reviewers over manuscripts and grant reviews and things of that nature. There are plenty of other wonderful things to do that are both intellectually rewarding as well as financially rewarding.
You have had a variety of roles throughout your career: mentor, director of various programs, chair of various committees. what are some characteristics or skills that you think have particularly helped you in these various roles, and how have you developed them?
Very good. Very good question. One is I obviously have no problem voicing my opinion or talking about things. Mark Gillespie, my PhD mentor at the UK College of Pharmacy, told me very early on. He says, "You're going to be all right because you have the gift of gab. You can talk to anybody about anything." Part of that comes from my kind of homespun farm boy upbringing in Frankfort, Kentucky. So when you come from a little small town Kentucky, not straight off a farm, but pretty close. You learn to just kind of have an all shucks, gee whiz kind of attitude toward things, and you learn to be humble. And so I hope I'm that. That's why I certainly strive to be. The ability to talk to people is a big part of it. And let's face it, scientists aren't seem to be known for that, although I think that's kind of falling by the wayside. I think the second and I hope again this is a value I try to live by, which is you have to care about people, and you have to be willing to give of yourself. And you have to, there have been times I have given a really hot project to a young investigator, and I said, well, go see if you can get that funded. It's yours. I got other things I can do. This is a pretty good one. I like it. If you're not going to do it, I am. But, I'll give you a shot at it because you're a young investigator and it's really hard. And so you have to be willing to give of yourself. You have to be willing to give of your time, of course. No more so than when it comes to mentoring. You have to be able to spend time with people and understand, where they're headed and where they've been and those kinds of things to be able to kind of give good mentoring advice. I think the other thing is don't be afraid to be wrong. I've been fortunate in that I was able to, I hit a bunch of grants very early in my career. That gave me a lot of confidence to try things. We've tried some things that didn't quite work, but I've always been comfortable in the positions I've had. I've always had good, caring people that I worked for, and they gave me the security when I didn't feel it to try things and just be willing to take some chances in my career. The other is I think you have to be able to work hard, play hard. That's always been my thing. So I have never been a scientist that burned a midnight oil. You will not find me in a science in my office or in the lab after 6 o'clock. You didn't find me as a graduate student. You didn't find me as a postdoc. You didn't find me as a junior faculty. Now there are times you have grant deadlines, right? And you'll spend spend evenings, weekends, nights, whatever, writing a grant. 2 nights ago, I spent the night in the sub lab picking time points up because I didn't want my lab to have to spend the night in the lab. So I said, hey, I'll go do that. I'll manage it. And it was rejuvenating. And so I think but I've always just worked really hard during my workday. But late afternoon, evening comes around, it's time to go enjoy life. And I play golf on the weekends. I used to hike a lot. I scuba dive. I fly fished when I lived out west. I do a lot of things. I've always had a lot of hobbies. I have 2 teenage boys. I coached 10 years of club baseball and club basketball and did those things. And so I was always able to manage a career and have all the other things in life. I think some scientists, discredit themselves by thinking that working 12 hours a day, 7 days a week for 20 years makes them a better scientist. I say it probably doesn't. And so I think people have to, learn that. But when I'm here at work, I work hard. I learned early on at 8:15 in the morning, there were tubes stalling in my ice bucket, and an assay was started by 8:30. And at 5:30, I was sitting down writing my lab notebooks, final thoughts, getting some overnight experiments set up. Used to have to do PCR overnight back in the Stone Ages and things like that. And so, just be organized. And if you do that, there's no reason you can't have a full life outside of science. I hope that kind of answers some of your questions there.
It does, definitely. I wish we had another hour, but I do want to be respectful of your time. The last thing I guess I'll ask is, what's a question that I should be asking and have not asked yet?
Oh, wow, what a good question that one is. Let's see. I don't know. That's the good one. You all of you asked some very good questions. Um, hmm. Well, you didn't ask me, what does it take to be a scientist in today's world? Or what's, oh, I know, a better one. What's different about graduate students today than when I was a graduate student? You didn't ask that one. Is it prompting me to answer that question? I'll put one out there. The thing that fascinates, I can't code. I keep saying one of these days, I'm going to take a 6-month sabbatical and I'm going to go learn to code. And it's just kind of one of these things I've never done in my life. I always, have used kind of plug and play kinds of applications and programs, especially when it comes to genetic bioinformatics kinds of things and what have you. Young people today, one of the things that I admire is many of them they just have no fear of computers. They don't have any fear of turning on some strange computer, booting it up, figuring out what it does, logging into this, logging into that, trying this, trying that, those kinds of things. I think that's great. Young people today, I have a very small Twitter, kind of footprint, and social media kind of footprint. I kind of begrudgingly do social media, just because, they all turn into just rants and raves after a while. So I don't want to get into that. But young people today are fantastic at networking, at self-promotion. Self-promotion is important in this business. You have to do it. So I think young people are much better at that today. I think that there's a number of things that that young people today clearly have an advantage over, in terms of, than say someone at my age in this career. I mean, I still sometimes have to question myself. Do I still do things the old way? Because I know the old way works. I don't know if the new ways are going to work or not. So, why would I try them, right? So that's where, having young people in the lab is great. One of my students just wrote an R code for doing the quick bioinformatics off of our drug screen assay. So we essentially get data analyzed as soon as it comes off the imager. The numbers drop straight into a program and it does the statistics. So it's almost in like real time the statistics are performed. And he wrote an R code to that. My 19-year-old son, who's a freshman at University of Arizona, just started to learn to write code, and he just goes on and on and on about R and all this. And I'm just kind of like, what's R? I know what it is. It's a computer code, and I've even looked at some of it. I even downloaded RStudio a week or two ago just to say I did. Those kinds of things, young people have so much advantage over today. So, that speeds research so much faster than it used to. So things can be done a lot faster nowadays. And I don't always have an appreciation for that. It takes you young people to come to me and say, hey, there's a faster way to do this now. Great, do it, please.
Thank you. Thank you for that.
All right.
I think we're out of time, but thank you so much for your time. You have been absolutely amazing. I hope we'll get to do a Kevin Harrod part 2.
That would be fun. Thank you very much. Anybody, I'm sorry we didn't get to interact with all of you individually, but if anybody wants to drop me a line, please feel free to send me an email. I'll be happy to answer any other questions.
And I did send everybody your CV, so, and then your email as well. So don't be surprised if you get emails.
Yeah, okay, sounds good.
Well, thank you so much.
Thank you all very much.
I hope you enjoy the rest of your day.
Great, thank you. Take care. Bye now. Best of luck to all of you.